First of all, thank everybody for coming. We appreciate the opportunity to present not only Dyadic, but more importantly, the technology platform that we spent the last 2 decades creating. That's a C1 technology platform. This is the safe harbor statement that basically says that whatever we say here today can change tomorrow. It's forward-looking, and things change, and with opportunities, good, bad, and indifferent, things can change. So we need to warn you of that, I guess. So the good thing about Dyadic is we're really not a startup company. We're a long history. I started the company in 1979. I'm not going to go that far back. But we have an experienced board and management team. We have people that come from Lonza, the people that come from Teva. We have people that come from Pfizer. So between the board and the management team, we have very seasoned professionals that have been involved in pharmaceuticals for a long time. And they all see what we see, that we have a very nice balance sheet of about $51 million in cash. So we aren't going to be raising money anytime soon. We hope never. And we have a plan that actually we want to execute on the next year and a half that still leaves us with $30+ million in the bank so that we can negotiate with big pharma with a strong hand rather than go with hat in hand. So it's all good there. We bought $19 million of our shares back last year, and we're buying another $5 million back, or at least have a share buyback program to do that. But at today's price, it's tough to get shareholders to fork up shares any longer. So it's all good. And we're listed on the OTCQX, which is the top tier of the OTC in the United States. And the reason we're at the OTCQX versus NASDAQ at the moment is we're really focused on science, executing on our business plan, and we infrequently come to conferences like this. We're more involved in industry conferences where we can try to trade partnerships, and you'll hear about some of those things coming up. So our footprint is global. We actually had an industrial enzyme business that we sold to DuPont for $75 million a year and a half ago. So we used to make enzymes and sell them in 35 countries around the world. So we are global. We have a global presence. We have a big presence here in Europe from a research and development standpoint and business development standpoint as well as in the United States. So what we're really doing here is we want to change the game in biologics. We believe that coming from the industrial biotech business, which is where we started and where we spent 2 decades, that the way proteins are made in the pharmaceutical industry are ineffective. They work, but they're not cost effective. You can use CHO cells, and they're safe and they're comfortable, and everybody likes to use those. But to be honest with you, we could never sell a product in the industrial world anywhere near the levels that CHO can produce, or Picky can produce, or baculovirus can produce. So we spent 20 years developing a workhorse, a production organism that can produce over 100 grams per liter of total protein with 80% purity. So we have examples of 80 grams per liter of this cell. And if you don't believe us, then take a look at who's using it. On the industrial side, we have companies like Abengoa, which you've probably never heard of because they're a biofuel company, alternative energy. They built a $500 million cellulosic ethanol plant based on this enzyme technology. Because without the enzyme technology, that plant doesn't run. They grew C1 up in 500 cubic meter fermenters. So it's 35 times the size of the largest CHO cell plant that Samsung built for $735 million. The beautiful thing here is we're going to scale down. We're not scaling up, but we can scale to any size anybody can imagine. In addition to, obviously, Abengoa, everybody, I assume, knows BASF, and you know DuPont. And of course, Shell Oil was a licensee through Codexis for the biofuel space. So this is something that works. It's been approved for US FDA for feed and food with grass, and now we're working on biopharma. This is a slide that just gives an example of some of the levels we've been able to achieve. And in total protein, we've actually exceeded these numbers. But in this case, where we produced an example here showing 100 grams per liter with 80% purity. That's 80 grams per liter in a 5 to 7 day fermentation. And that's versus the world record of CHO is 10 or 12 grams per liter basically in 14 days, not counting the fact that the media cost is 25 times more expensive for CHO and we don't have viruses. You're talking about if we can get a fraction of that yield and the time savings we have, we're talking about really making a difference in the biologics development and production. So the goal here is to take C1 into the pharmaceutical world. We're a year plus into that. We've got 2 pharma relationships with 2 of the top 10 pharmaceutical companies currently funding research. So not only do we have our own capital, we're getting some funding from the big pharmas, and we're hunting down more of that, because actually I own a third of the company, so I don't like to spend my money if I can use someone else's to advance the platform. So it's very good from an investor-friendly base. So C1 for biologics. The opportunity here is the world has spent billions and billions of dollars up front on genomics, on proteomics, on discovering genes, on changing the properties of the proteins that these genes encode for, but they virtually have left the biomanufacturing process the way it's been. So it's like driving a Model T car in a Tesla world. And so now we're taking synthetic biology and advancing C1 by genetically engineering the cell in vivo so that it can produce proteins that look like they're human. So in addition to being able to make the proteins that we can make today, we're modifying to be able to make vaccines, to make antigens, to make monoclonal antibodies, to make bispecifics. And the breadth and the scope of this is still in development, but you'll see we've already succeeded in doing a lot of those things already and are on our way. So this is just kind of an article series that came out from Biopharma Reporter from Dan Stanton. I don't know if you guys get this, but if not, it's online. It would be good to read. And his first article came out in, I think it was April of 2017, Showstopper, Biogen looks to alternative cell lines. And the point here is that the pharmaceutical industry is waking up, and they're realizing now that CHO may be good, it's safe, it's effective, they're comfortable, but they need a better mousetrap. And so in this case, and I guess it was August, 4 months later, they put out an article about Dyadic and C1 potentially being the showstopper, that we have potentially something that can actually revolutionize the game. And then recently, last week, they came out with a survey where they said that 45% of the biopharmaceutical executives and the biomanufacturing executives think that we're too reliant on CHO. Of course, we agree with them, and that's why we're doing this, but it just shows you that the shift is finally coming, because in the past, they didn't care about cost of goods, but with healthcare and managed care and the way the payers are squeezing all the prices, it's coming down. And what people are doing today with biosimilars, they're not really improving the manufacturing process. In fact, the guys that are making biosimilars are probably producing it for more cost than the brand, because the brand's been doing it for 2 decades. They're just cutting margins. So they're not really doing anything but just cutting profit margins. So we're actually improving the manufacturing process. And that's going to be the second wave. So what's the science behind the curtain? C1. So why filamentous fungi? Well, first and foremost, filamentous fungi in nature secrete proteins naturally. So that's good, where CHO cells were forced to do that. They produce very high levels of proteins in nature, but typically they produce them in a very viscous manner and in a way with not high purity. And so through serendipitous mutations and random mutagenesis and good genetic engineering over 2 decades, we've changed the morphology of C1 so it grows like a yeast, unicellular. So we took care of the viscosity, brought it down from several thousand centipoise to less than 10. So you're stirring up water versus oatmeal, big difference. That's why we can scale up to 500 cubic meters, for example. But at the same time, what happened is these long filaments, which are really microscopic, but compared to yeast, they're long filaments, became micronized through a random event of scientific anomaly, and that gave you hundreds of tips in the same mass. So you're spewing out protein from 100 tips instead of 2. So the yield went up, the viscosity went down. We continued to do mutagenesis to C1, and we had another sort of scientific anomaly where we just wiped out the secretome. If you see those protein gels, that gel on the right of C1, that's not purified protein, that's how it comes out of the fermenter, versus having all these protein bands. So in the past, fungi would produce 50 different enzymes at once. So you put in a protein or gene in that system, you're competing with all the other energy it's using to make the other proteins. So we wipe the background out. And that makes purer protein. So we can produce higher levels in shorter time in purer form under wider operating conditions, pH and temperature, at much higher yields than CHO cells. So we also have the genetic tools, which we had to create from scratch, because of course, unlike E. coli, for example, you can buy a kit, or Picky, you can buy a kit. We were the only ones that were working with this beast. So we had to develop the secretion signals, the promoters, the vectors, the transformation protocols, all from scratch. And we have all that. So not only can we knock genes in, we can knock genes out. And that becomes important because, of course, cells make proteases, so you want to get rid of that background activity, and we have the ability to do that. So this slide is a slide demonstrating that we've now taken 4 different mAbs, monoclonal antibodies, and produced them. And they're biologically active, and you'll see some things coming up on that. And we now have demonstrated that we can produce mAbs within a very short period of time, much shorter time than you can with a CHO cell, in 2 to 5 grams per liter in half the time or less. So if you had a CHO cell producing 5 grams per liter in 14 days, they'd have to be at 10 to be equal to this already. And again, we do this with 1/25th the media cost, virtually salt and glucose, versus the way that CHO cell, very expensive media, and you skip 2 viral filtration steps because we don't have viruses in C1. So not only can we make more, we can retain more through purification and filtration. And so we have all the tools and are using those tools to genetically modify the cell. You'll see about glycoengineering, where we're then modifying the cell so the proteins come out looking human. So we can do vaccines today and stimulate immunogenicity, and now we're working on getting rid of immunogenicity through glycoengineering. And that's not something that people look at and say, well, can you do it? It's already been done in other systems, like Pichia. You know, Glycophyte did it and got paid $425 million from Merck back in 2007. The problem is they did it to a yeast that wasn't that productive in the first place, and when they got done glycoengineering it, it was no better than CHO, so who's going to use it? This is like Glycophyte on steroids, and that's the goal, where we can have more consistent glycosylation in the end with much higher yields. And we're not stopping here. These are slides showing advances of potential synthetic promoters to drive yield even higher. And so we're working on new modern tools. We're not standing still. Generation 2.0, generation 3.0, et cetera. And here's an example of a monoclonal antibody that was expressed with one of these synthetic promoters. And you can see from this Western how much brighter those gels are. This slide basically shows you that we've compared now a monoclonal antibody produced by CHO to one that's produced by C1. And the binding is virtually identical. And that's important. And so we've now reached that milestone. Glycoengineering. So this is sort of the last step, although there's interim steps of other things we're doing, but this is the major step, is can we take C1 and modify the glyco pattern of the proteins coming out of it? And the answer, we believe, is yes, and we've begun that already. And we've actually, in the first step, dramatically changed the glycosylation structure. But if you look at C1 to start with, it's much simpler. It's much closer to human because you're going to glucanac man 9, not man 23, man 35, et cetera, that you'd see in Pichia and yeast. So we're much closer to start with to human, and we're moving it down. And the interesting thing here is we're going to end up with hyperproductive cell lines with a variety of glycoforms. So you can actually put your gene into any one of those versions at the same yield and see the benefits: higher immunogenicity, less immunogenicity, no immunogenicity, and on top of it, maybe better performance, because people have not been able to do that. One of the things that we haven't done yet, or we've done but haven't shown here because of time, is we've actually made vaccines in C1 as well. And not only did we make them cheaper, we made them equal, if not better, immunogenicity. So this is just a timeline. So if you give us a gene, we can do the synthesis of the gene, the construction of the gene, the transformation of the gene, create cell lines, produce the cell line, pick the winners, the higher producers, put them in a fermenter, purify them through protein A, and do analyticals all within 15 weeks or shorter. So that's a much shorter timeline. So not only can we do that, when we produce them, we can save at least a week in the fermenter, and potentially a week before the fermentation. So it's quicker, faster, cheaper, lower media costs dramatically, no viral inactivation. And then the CapEx. That $735 million plant that Samsung just built might have been a heck of a lot cheaper if they used C1. So this is the future. We're heading towards it. We can't fine-tune a Model T, which is CHO cells, so we're using the Ferrari or a Tesla engine and driving it forward. Shorter, higher yields, all the things we talked about. And we're out of time, and thank you very much.